Thanks for the opportunity. It's great to be here. I hope that we all together can convey today some hope. It is a good time for the field. A lot of the things that we've learned in immunotherapy have been paved by what we've learned in melanoma, especially skin melanoma and other diseases. But we do believe that the years to come will be an opportunity to systematically try to understand well why skin melanoma responds so well, why uveal melanoma doesn't respond so well, and try to build upon those concepts to then empower the ability to treat the disease with what, in my opinion, could be a potential cure, because the responses that we're seeing in skin melanoma, these are long-term durable responses like we've never seen before in the field of medical oncology. So if any of you provides or volunteers to provide a drop of blood and we look inside, then we'll find that there are thousands of red blood cells floating around, but there are also thousands of white blood cells, literally white cells, some of which are T cells or T lymphocytes. These are essentially the cells that are constantly patrolling around your bodies, looking for invaders, infections that will eventually destroy them and clear the body from an impending infection. So if you model this in a computer through a specialized microscope called a scanning electron microscope, you'll see a killer T cell, which is really the leading cell within the immune system that can actually engage a tumor cell and engage in a manner that recognizes that tumor cell in a very specific way that can eventually eradicate and kill the cell completely by releasing specific chemicals that will eventually destroy the cancer cell. Now, as a general concept, immunotherapy is a treatment that uses your body's own immune system to fight cancer. And this could be accomplished in different ways. I'm going to try to specify this a little more at the end. But in general, as in life, there are good players and there are bad players. So the good players of the immune system are the killer T cells, the one that I showed before on the previous slide. And then there are also bad players. And those bad players are really conformed by different types of cells within the immune system that I'm not going to specify in detail. But those cells have the ability to slow down or interfere with the process of the good players, the T cells, in terms of destroying the tumor. So at the end, what we want to try to accomplish is it doesn't matter how we do this, and there are multiple ways to do this. Somehow, the panel that was before me had described different clinical trials that are actually about to happen or happening in terms of how we can modulate the immune system, how we can manipulate the immune system to our advantage and destroy cancer. But the general concept is, let's empower the good players and let's try to get rid or minimize the effect of the bad players. So the question is, how do T cells recognize tumor cells? And really, I've tried to make really this very simple, and the only medical term that I'm expecting you to learn today, it's the word antigen. So essentially, on the upper part of this slide, the T cell, the killer T cell, has a receptor or reveals a receptor on its surface. And that's called the TCR or T cell receptor. But we'll keep it simple. It's really just a receptor. Now, on the other end, the cancer cell serves on a platter, and the scientific name for that, it's a MHC class molecule, but let's call it a platter, where the tumor somehow presents or serves part of its own identity. That little orange circle is just a minute part of the tumor. It's matter, essentially. And the scientific term for that, it's an epitope or an antigen. But essentially, that antigen there can somehow bind to this platter in a very specific way, therefore providing and allowing an opportunity for that T cell to match as a lock, as a key within a lock, in a very specific manner, and be able to discriminate among thousands of other cancer cells that are presenting other completely different antigens, but in this case, the T cell binds in a very specific manner. Now, if the T cell does this, can kill and destroy the tumor cell. And if you look at the bottom of the slide, when the tumor cell dies, then what's going to happen is that now all this minute pieces of matter or antigens are just going to spill outside of the cancer cells and are going to be taken up by a different cell that's called the antigen presenting cell, or the APC. So this cell, it's a specialized cancer cell. It's a specialized immune cell. You can see all this dendritic slender projections like a star, which, essentially what it's trying to do is trying to have the ability to capture and uptake all of this antigen that's being released around, to eventually, again, using a platter system, present it again to an incoming T cell on the far end. So when that happens, now the T cell has the ability to essentially recognize where the tumor is. And what's going to happen is that it can now turn around and go after a tumor cell and eventually kill the tumor cell. So this is, in simple terms, how the immune system is able to recognize and kill cancer cells. Now, this is a patient with skin melanoma. Now, what happens if we look inside a patient with skin melanoma? Well, this is a tumor that's been growing for a while. And if you look inside this tumor, you'll see thousands of cells. You'll see thousands of tumor cells in blue, but you'll also see several T cells in red. And there are other cells that I've shaded in gray here because, for the sake of simplicity, are, again, cells, some of which can be good players, some of which could be bad players. But essentially what happens is that if we take this tumor that has all this complexity, all these types of cells, and we chop it into small pieces, literally very small pieces, and we take those small pieces and we put it to grow in the lab on a special plate that has special chemicals, that chemical essentially what allows is that the T cells that are part of those small fragments can preferentially grow, multiply, and divide into billions of cells. So when that happens, those cells that have expanded in vitro, meaning in the lab, then we can infuse them and give them back to a patient where we have previously reset the immune system using chemotherapy or radiation. So we reset the immune system with chemotherapy, radiation, so now to open space of the incoming T cells that we're hoping that can go after the cancer and destroy the cancer. So over time, essentially, the T cells can accomplish that. And over time, this tumor that was well established eventually completely disappeared. And this is really courtesy of my colleagues from the National Cancer Institute, where this mode of therapy called T cell transfer immunotherapy was actually really pioneered. And what this really proves is just the concept that there are specific T cells that can recognize cancer cells in a very specific fashion and can go after the cancer and destroy up to the last single cell. So we know that the capability is there, but this doesn't always happen all the time. Some patients achieve these remarkable responses, but some patients just don't respond at all. And that's where it gets very complicated. The notion that the immune system could have been relevant or could be relevant in uveal melanoma came from really decades ago when a group of doctors in Europe really reported about a few patients that had developed uveal melanoma, but eventually the tumors disappear on its own. And the question is, how could that happen? I mean, how come a tumor just disappear on its own when it's been established there for a while? So the notion was that there's possible that really there's an immune system that can recognize the cancer and just without any type of manipulation go and destroy the cancer cells that have grown. So this was really the first observation to suggest that there was a role for the immune system in uveal melanoma. And the question is, how much interaction do we see in uveal melanoma between established cancer and the immune system? Well, it's been shown that indeed T cells are present within uveal melanomas. About 20% of uveal melanomas in the eye, this is in the eye, really have T cells, and the T cells just hanging out there inside of the tumor. And it's clearly that as the tumor grows over time, there are more T cells immersed in that cancer. This has also a correlation with the amount of blood vessels that tumor has actually been able to develop over time. And unlike other cancers, when we see T cells in uveal melanoma, we really don't think that this portends a good prognosis, but instead might be detrimental. So if we see T cells that are there, why aren't they doing their job? So the thing and the problem is that these T cells can be completely paralyzed, neutralized by the cancer cell. We can somehow find ways to try to, again, reactivate these T cells. But the problem is that the tumor cell, it's very, very smart. And on the surface, that tumor cell, which is shown here in yellow, can now reveal, let's call it a marker or a safety checkpoint that can eventually bind very specifically to another protein that's actually expressed on the surface of the T cell. Now, that engagement of those two proteins up there, it's a safety checkpoint. Essentially, the cancer cell senses that recognition by the immune system and says, this is not looking good. So what it does is that it can actually neutralize, paralyze that T cell, therefore it cannot kill the cancer cell. Now, in the bottom panel, you see that once the tumor cells are dying, as I said before, all these antigens can be taken up by APCs, antigen-presenting cells, again, presented to an incoming T cell, but again, the same type or similar type of engagement will occur here, where a blue marker or a blue protein revealed on the surface of a T cell binds in a very specific fashion to the antigen-presenting cell, and again, that essentially functions as a safety checkpoint, and it really neutralizes or paralyzes the effect of the T cell. But nowadays, we've had what we call checkpoint inhibitors, and this is something that you'll probably read about a lot, that these are monoclonal antibodies that can essentially go now and interfere with that binding between those two safety checkpoints, to the point that when that occurs, now the T cell can be reactivated and kill the cancer cell. Same thing with a different type of monoclonal antibody. We can accomplish the same strategy by blocking the recognition between an antigen-presenting cell and the T cell. So these two modalities of treatment nowadays, you'll hear about Yervoy or ipilimumab, Opdivo or nivolumab, Keytruda or pembrolizumab. These are checkpoint inhibitors that have been now approved to treat skin melanoma and other diseases, other types of cancer, that are now paving the way as probably one of the most clever and elegant strategies to reactivate the immune system within the body of a patient. So when that happens, then this T cell down here can essentially go back, be reactivated, and then bind to the tumor cell and completely destroy the tumor cell. So how does this translate into patients? So when we treat patients with skin melanoma, this is really one of my patients that I treated with a combination of ipilimumab and nivolumab. This is skin melanoma, and all these little black spots, this is all metastatic disease throughout the body. And after 12 weeks of treatment, then you see that all those black spots have almost completely disappeared. Again, proving the concept that there are T cells inside of those tumors that can be reactivated to then go and destroy the cancer. But this is skin melanoma. Now, what happens in uveal melanoma? Well, this slide is a courtesy of my colleague, Dr. Lin Fun. He treated this patient with a combination of, again, ipilimumab and nivolumab. So patient presented initially with this metastatic site in the liver. This is a CT scan, and this is the liver here. And you can see this area here. It's the metastatic deposit. The patient was treated with 4 doses of the combination and then 19 doses of nivolumab alone. And you can see that there has been some degree of response over time. And the problem is that this type of responses are only achieved in a subset of patients with uveal melanoma. They're not as profound and as striking as the ones that we see in patients with skin melanoma, And the question is, why is that? So one of the things that Dr. Yude Kamula at the NCI has been trying to study now for a while is that he actually asked this question in the lab, and he looked at metastatic deposits from patients with uveal melanoma and compared that with patients with skin melanoma. And what his group found is that essentially the patients with skin melanoma had T cells, had much higher number of T cells that were able to recognize the cancer cells and then become activated, okay? That means not only one T cell expressing one receptor, that probably means several T cells with different receptors recognizing different antigens on different cancer cells. But instead, when they looked at uveal melanoma patients, they saw that there was really not a lot of T cells that were actually recognizing the tumor. And probably because the population of T cells there had receptors in purple that would have never matched the orange antigen. So the conclusion of this study is that the reactivity or the ability of T cells to recognize cancer cells in skin melanoma, it's much higher than the ability of the T cells in uveal melanoma to recognize the cancer cell. A lot of speculations, and these are all theories, and it remains a matter of investigation, that we presume and we think that there might be a role that perhaps the amount of antigens that are expressed on the cancer cells are much, much more than the ones that are expressed on uveal melanoma cancer cells. But actually, when they looked at this in the lab, they really don't see a clear-cut correlation to really conclude that that's necessarily the case. So the story may be much more complicated than that. Now, but what happens is really the eye is this complex but marvelous anatomical structure in humans, and naturally has evolved in a way that it's built in itself mechanisms of immunoregulation, or mechanisms that in the setting of physical injury, physical trauma, or let's say infections in the eye, the immune system can get activated, but at the same time it gets activated in a way that will try its best to clear the infection, but at the same time, not to a high degree that's going to compromise vision itself, because vision, it's really, for every human being, it's probably one of the most important senses. So this is something that naturally has evolved that way. And there has been a lot of work done in the past about what happens if you artificially inject antigen into the eye of a mouse. And if you inject antigen, meaning foreign material from different sites into different areas of the eye, then what happens is that you can activate the immune system. But when you activate the immune system, you're activating and you're inducing a lot of what we consider, again, in tumor immunology, which is a different scenario, a lot of the bad players. And the question is, if this is the way how naturally the eye has evolved over time, is it that some of this built-in immunoregulatory mechanisms are somehow carried over when we have a cancer that's trying to develop itself in the eye? And the same way the immune system responds, it responds in a way that it essentially allows the cancer to establish itself, to shield itself, not to be seen by the immune system and then eventually metastasize to the liver. And the answer is, we don't know. So the promise of immunotherapy really will rely upon a very systematic approach in terms of how do we understand the biology of this cancer. And some of the investigators in this room have actually alluded and specified about different immunotherapy trials that have really now are about to open, that are actually happening. And we'll see which of this actually eventually lead to optimal responses. And we remain hopeful that several of this will do and will accomplish that. But the points where we can intervene are essentially the following. So the first component of the response, as I said, it's going to be the T cell. So we have to make sure that the qualities of the T cells that we have inside the tumors are really those T cells that are armed and capable to destroy the cancer cells. And there are different flavors of T cells. Some T cells are better equipped. We can actually grow some of these T cells in the lab by exposing them to different chemicals. And by doing so, the T cells, some of which will become more effective compared to other ones in terms of treating and killing cancer. So not only the quality, but how diverse the T cells are. How many T cells we have that can recognize multiple antigens expressed on a cancer cell, it's really one of the main limiting factors. Now, one of the ways that this could be accomplished in the clinic, it's by essentially trying to vaccinate patients with cancer in the hope that these vaccines will eventually now increase the trafficking of all these T cells around the tumor. Now T cells that are much more diverse, that can recognize multiple antigens on multiple cancer cells, that can eventually be mobilized by using other strategies than to destroy the cancer cell. So another way that this could be accomplished is by essentially genetically engineering the cells in the lab by introducing genetic material that will then code or make that T cell express a receptor that's specific for a specific antigen, like the MART1 story, for example. And this is something that can be done in the lab. And with this approach, essentially when you do this, the T cells that are produced are usually derived from patients. They're manipulated in the lab, and then billions of cells. You need billions of cells to be introduced back into the patient to be able to go and fight that amount of tumor that I showed you before. Otherwise, we may not see that many responses unless the T cells have a much better quality where you don't need that many T cells, but instead the quality of the T cells is so good that they can still accomplish the same goal. Now, another way that this could be manipulated is that using specific drugs to make sure that the tumor cell serves those antigens on the platter. The problem is that the tumor cell is so smart that it can actually hide from the immune system by hiding this molecule here. And if it hides that molecule, then the tumor cell cannot be seen by the T cell. So there are ways that we are studying in terms of how to manipulate this tumor cell by exposing the tumor cell to, for example, epigenetic modulators, to try to increase now the amount of antigen that's expressed to potentially raise visibility and have the T cell destroy the cancer cell. Another way would be, as we said before, by using this specific checkpoint inhibitors or antibiotic drug conjugates, I was actually alluded before, that will essentially, in a very targeted fashion, will take that specific chemotherapy drug into the cancer cell, and then once the cancer cell takes and uptakes that chemical inside, it'll be delivered and it'll kill the cancer cell. So the other thing that we have to do is essentially get rid of the bad players that are constantly putting this negative effect on top of the T cells. So there are ways to accomplish that, not so effectively in the clinic yet, but we are starting to see the possibility of really getting rid of the bad players, not so much of the cells itself, but in inhibiting or depleting or minimizing the effect of some of the chemicals that these cells produce that can have a negative impact upon the T cells. Another way, again, it's by increasing the repertoire or the diversity of those receptors on the T cell to recognize multiple antigens, and in a different way, not only make sure that the antigen is expressed, but it's expressed in very high numbers. So one of the work that we're actually exploring in Dr. Bill Harbour's lab, and this is work that was published by Matthew Field from Dr. Harbour's lab, it's essentially that they've discovered a gene called PRAME, and that he actually discussed before, which is really a protein that's expressed, like down here, on uveal melanoma cells that really helps to prognosticate and stratify patients into two groups. Those patients that have cancer cells that express high levels of PRAME and cells that express low levels of PRAME. And we're starting to see that those patients that actually express really high levels of PRAME behave differently than those that do not. So PRAME, it's really a protein that was described in the past to have the ability to trigger an immune reaction. So it can do that if it's really presented by the cancer cell on the appropriate cervical bladder. It can do that probably if the amount of the antigen is reasonable that the T cells can see it, but we are starting to explore different ways of how to manipulate this or immunomodulate this to the advantage of trying to get rid of the tumor. So this is really essentially a summary of the clinical trials that in a way have been discussed so far. It's just only to exemplify that what's been explored now in the immunotherapy field encompasses a combination of different strategies. For example, on the top trial, the ability that we have to genetically change those antigen-presenting cells now to make them express high levels of a specific antigen. It's a way to vaccinate patients and try to stimulate a specific population of T cells in the body. Dr. Sapna Patel actually discussed the combination of nivolumab in combination with ipilimumab at MD Anderson, which is again combining two checkpoint inhibitors we discussed before. And we're eager to see what the results of this trial, one of which is also being now performed in Spain, will eventually lead to how many responses, and if indeed we're going to see the same amount of responses that we see in skin melanoma. The third trial, it's really essentially a variation of number one, but it's again, these dendritic cells or antigen presenting cells, which are essentially the same, are being loaded on the surface with RNA or genetic material that's been really derived from cancer cells, in the hope that these dendritic cells will then be able to stimulate an immune response. Then pembrolizumab in number 4, which is another checkpoint inhibitor, it's being tested as well as a single modality. We don't know if pembrolizumab alone is going to be as good as a combination of nivo and ipilimumab. We do know from skin melanoma that the combination of nivo and ipi actually produces much more profound immediate responses, but it also produces much more toxicity, and we don't know how this is going to pan out long term. Then there are other strategies like in number 5, where it's been discussed before with ICON-1 by injecting this inside the eye, or with IMC-GP100. IMC-GP100, based on the slide that I showed before and what Dr. Lutsky actually had alluded before earlier, it's a way to then try to engage T cells that have existed somehow there before, but now are being engaged and are being brought near the cancer cell by using this linker, or what's called really an immune mobilizing monoclonal TCR against cancer, which is the IMC-GP100, so that the T cells can be brought close to the cancer cell and really destroy the cancer cell. So this is really a very elegant approach in terms of the fact that the TCR has been genetically modified to have a lot of affinity for its antigen, and it may be the case that it can actually do that and bring those T cells and cancer cells together really effectively. But we'll see long term. So far, a signal of response, but not the degree of responses that we would love to see. So in terms of number 7, pembrolizumab with entinostat, again, it's really combining somehow medications that can change the phenotype or the properties of that cancer cell to facilitate recognition by the tumor cell. Glemba was discussed about before as well, with an antibody drug conjugate. The carbazine, which has been out there for years and was really the only chemotherapy, one of the first chemotherapies we had to treat skin melanoma, it's now being combined with interferon alpha 2b, which is really a medication that we've used before. Interferon alpha 2b, we don't know precisely how it functions and how it can treat cancer, but it's felt to have really diverse ways of acting or immunomodulating the immune system. So this is again another form of combining immunotherapy with chemotherapy to try to improve the outcome of immunotherapy or chemotherapy. And last but not least, this is Dr. Yuday Kimura's work at the NCI, where Immunotherapy relies on the use of tumor-infiltrating lymphocytes, essentially what I showed you before, dissecting, chopping these tumors into small pieces, growing the T cells in the lab. Also work of this nature has been done at MD Anderson by Dr. Pat, who in skin melanoma. But this is essentially trying to exploit the ability of using the T cells that have pre-existed there for a long time now, to grow them into billions of cells in the lab and then infuse them back into the patient and see if they can accomplish the same degree of responses that we've seen in skin melanoma. So far, we haven't seen the same degree of responses, but we remain hopeful that as we can recognize those patients that can potentially respond to immunotherapy, that we'll see better responses. So I want to thank you all for your attention, and we'll take any questions if there are any questions. Thank you for your time.
Any questions? Maybe 2 questions maximum.
When you talk about patient responses, are you talking about complete responses, or are you talking about stable disease or decreased disease?
So actually, with T cell transfer immunotherapy, again, in the setting of skin melanoma, we see that you need to achieve, if you achieve a complete response, most likely you will be cured. The patients that can achieve a complete response have been alive long-term for more than 5 years. This is in skin melanoma. Again, we don't see the same kinetics of response in uveal melanoma. With the checkpoint inhibitors, what happens is that there are some patients that have profound responses where everything goes away. There are patients that have only partial responses, and then the disease stabilizes and doesn't grow anymore. And there are patients where essentially you see a modest change in the size, it shrinks a bit, but then it remains stable, but it can remain stable over a long period of time. So we don't know how to predict for that. We don't know what type of response a patient will have, but even stable disease, for example, with checkpoint inhibitors, like let's say the patient treated with ipi/nivo with uveal melanoma, that could be a long-term benefit, and only time will tell.